And I'll just so we're recording it for those of you who want to save this for posterity. Now let's give it a couple more seconds to get started. All right, why don't we dive in? It's just going to be Maria and I talking to you all today. So today we want to, so everyone can see my screen. There we go. All right. So we have a new resource on the horizon, and I wanted to share it with you all. We haven't actually deployed it yet. It's a preview, and today I want to talk to you about it specifically as our user base in complex trait genetics and genomics. I really want to talk to you about it because I think it means a lot for our current established resources. It also will bring a new resource to our community, but it will enhance the current existing knowledge portals for common disease, for common metabolic disease, and for our A2F portals. And really what I want to hear about from you all today as we have a discussion is, any ideas you have as we share with you our thoughts about what we're embarking on that may be useful for us to bring into the knowledge portal. So I'm going to give you a little setup today. I'm going to talk to you a little bit about what we're planning to do, but I really want to, and then Maria is going to actually review parts of the resource that are really cool that we're thinking to bring together. And then hopefully we'll have some Q&A, but feel free again, pop things in the chat. So I actually want to begin with something that is a funny question, but maybe worth asking. Does anyone on the call know what the Common Fund is? And have you used any of its resources? If anyone wants to answer that, that would be fantastic. Oh, Steve has raised his hand. Steve, what have you used? Can you unmute yourself?
There you go.
Hey, there you are.
Oh, can you hear me? Yeah, so I've been doing some research into this to learn a little bit more about different subsectors of the Common Fund, but essentially it's you want me to talk about it a little bit, or what would you prefer?
I would just more like just a quick, like 2 sentences, what you know about it, and then have you used any of its resources? Yeah.
So I haven't used the data sets as such. We've actually been going over what is available in the Common Fund, but the Common Fund are generally larger data sets that are generated from specific subfields that are funded by mechanisms outside of a particular NIH institute, if I remember correctly. And the reason why is because sometimes they're a little bit more they have a slightly broader scope, or they have there's some issues about like how how much risk there is with the thing. So I believe this is a lot of the idea. You're generating these enormous data sets for these functional questions, like, say, for instance, nucleosomal structure or glycosylation. There's all sorts of different ones, GTEx, for instance.
Yeah.
So yeah. So, but
That's
yeah,
great.
sorry. Okay, cool.
That's great. Anybody else? I know I saw someone, you've never known this. Well, that's great. That's a good thing. You do research. So hopefully some of the resources that they have in the Common Fund will be useful to you. And that's really the hope. Our hope is to raise awareness about these resources. Some of them are incredible. And I'll tell you why I think they're incredible, because we've done some research over the last 6 months and I've learned a lot. And I really want to talk to you about it today because I think it's going to be really fun for our portal. So quick, Steve, you did a great intro for us. The Common Fund is really an audacious platform at the NIH of the Office of Strategic Coordination to specifically fund kind of bold, large-scale data generation and integration efforts across science, across the NIH programs and missions. And there's a host of these programs. I'm not going to go into all of them today. They're really impressive. And they kind of have 3 major pillars. The first pillar is transformational science and discovery. So these are programs designed to have new models, new resources to basically have new pathways or new scientific discovery. So like really sort of a specific area of science, generating a lot of data in that space and creating a foundational resource for that. The other pillar is re-engineering of how we work. So how we empower people, educate people, think about the actual patients on the other end of the spectrum, and promote equity in our research. The third pillar is our catalytic data resources. So they sort of leverage all 3, they pull it all together, they really are managing big, large data warehouses and make those resources accessible. Then there are these translational projects that cut through them. But the goal of these is to really span science across the NIH institutes and also feed into the various disease-specific interests so that they can basically rise up all divisions of NIH. It's a really exciting program. It's been around for a long time. So many of you might have used some of these and heard of them. GTEx is one that I bet you a lot of you have used, right? So the reason this is really exciting is because about 5 years ago, inside the Common Fund, they wanted to find mechanisms to raise awareness about the Common Fund resources. So they took 14 or so of the resources and they decided to develop a consortium within it called the Common Fund Data Ecosystem. And their goal was strictly to enhance the ability to ask scientific questions across these data sets. So they did this by funding partnerships. So saying to, if you want to work with 4DN, which is one of the resources, and if the resources themselves want to find ways to work together, they can have themselves create partnerships. And the Common Fund was funding sort of these partnerships. They also had these RO3 mechanisms and other scientific RFAs that went out. But moreover, they really wanted to build a way for everybody around the ecosystem to know what the other resources in the ecosystem are doing, but moreover, make them accessible to others. So this landscape is huge, in fact. This just gives you some work that one of the resources that was funded in the early days of the pilot to just sort of survey and create a catalog, if you will, of all the Common Fund data sets that are out there in these 14 resources. And they did an incredible job of cataloging all the data from the different types of data types and the different types of assays or entities you can access. That was amazing, right? That was the first effort. But then they were like, well, wow, we have such rich resources here. We need to make them accessible to others. And so we thought of a great opportunity for us. Well, you all know we aim to build portals that simplify knowledge access, right? We do this for human genetics and genomics pretty well, I hope, right? And that's a very specific focus. It's a lot of different data types, right? Genetic data is one major type, but genomic data is a really large span of data, but it's really trying to answer specific set of questions. What is the human genetic support for my favorite disease, right? So it's a very simple model. But we've been really excited over the past couple of years, doing these portals. We've heard along the ways about all these other resources that are doing incredible things, generating data in different spaces. And as you know, we've incorporated some of them into our resource, GTEx being one of the Common Fund one, right? So if you want to search on the portal for your favorite gene, you will actually get the bulk gene expression for GTEx represented on our portal as distilled knowledge from that resource in the context of our resource, right? So that's kind of how we do this. But we want to sort of take that one step further, right? And we do this in the context of working directly with the community. So these are disease-specific communities. They understand the disease. They understand their scientific questions they're trying to ask, right? So we want to take that same paradigm and kind of think about how we would do that for all these Common Fund resources. They themselves are sometimes 7 or 8 years old. They themselves have their huge communities of people who are actively generating data, participating in consortia and other funded entities, writing papers, making scientific discoveries within their resource. But they are kind of their own club, right? Just like all of our disease communities are on their own club. But if you could find a way to work across clubs or to distill information from those clubs to other communities, you might actually learn more about biology. Also, what we enable with our portals is we try and make things easier, right? There's a scientific process that all of you go through when you are trying to discern whether you want to pursue a particular loci for disease. Without the knowledge portal, you have to go to all these different sources, right? You have to know R, you have to know how to download, you know how to find things. It's exhausting. It's doable, and if you're an expert, which many of you are, you can do it, right? But wouldn't it be great, right? If you just go to the knowledge portal, you type in your favorite gene, you can get all those data sets and results distilled for you easy, but you know that the process behind it is that process you would have had to do by yourself. So that same paradigm, we also want to translate to this knowledge center that we're trying to build with the Common Fund. So we want to take that same philosophy, right? Collaborating directly with the resources, onboarding pre-computed results from those resources, incorporating all of them, encoding analytical methods from these resources that are best practices, and making those integrated and accessible on a portal. But it's not just enough to do it exactly the same way, right? We have to totally step outside our comfort zone for this particular resource. So we want to take our original vision, right, for the things that we've done well in the past, the Common Metabolic Disease Knowledge Portal, the A2F portal, the Biomedical Translator, where we're building these very large knowledge graphs that many of you are aware that we're participating in, and the fact that the Broad, we work very closely with the Broad team that builds Terra, which is a cloud-based infrastructure. I'm seeing a question in the chat. I'm going to pop it. Okay, great. That's good to know, Eva. All right. All right. So with that, what are we going to do? So we were luckily included in this ecosystem for its next round as a funded entity to build a common fund data ecosystem knowledge center. And the goal will be to empower the research community to use common fund data sets for novel scientific research that was impossible before. And we hope this will include hypothesis generation, discovery of new validation for targets that lead to better insights for human health and disease. So applying it to health and disease really across these 14 or so resources. So what are our aims? What are we planning to do, right? We want to work across all these common fund resources and the investigators to define a knowledge model. And I'll talk about what that means in a moment. I want to implement and extract knowledge from all these. I'll talk about that also in a moment. This is a lot of work. This is a 5-year effort, right? We want to develop a user-friendly knowledge portal, similar to what we've done historically, but really tailored to a totally different user base and scientific questions that are quite different in scale. We're hoping to work very closely with our the other common fund data resources to perform outreach, because they have their own outreach, but we want to have outreach that really promotes this ecosystem of all these resources that are coming together to share knowledge. And we want to work with all the other funded entities. So that's kind of our aim. So let's talk a little bit about what we're going to build. So you may know GTEx very well, right? You probably go to GTEx for your favorite gene and search for bulk tissue expression across all of it. So here's an example for FTO. You see the violin plots. That may be one way you use it, right? So this is sort of like the classic way someone uses GTEx. And human genetics is incredibly relevant. You want to know expression of your favorite gene if you believe it's implicated in disease. So this gives you an example of a common fund resource that we are incorporating into this knowledge center that you're quite familiar with, right? Here's an example of one that I had no idea existed. But I got to be honest, after reviewing it and watching it, I was both humbled as someone who builds knowledge portals with our group as to what tremendous resource and presentation of results are accessible in these resources. So this is Spark. This is a resource that builds maps about basically the electrophysiology of signaling in our body. It's crazy, crazy cool stuff. And the visualizations and the tools are incredibly rich and incredibly well curated, well documented, beautifully extracted into these visualizations. This is amazing. I bet you didn't even know this resource would exist. As a human geneticist, you might not actually ever use this resource, but we're hoping as we work with them to understand the data that they've generated. In many, many of these resources, they've generated omics data at scale, major scale. They have image data. They have these very cool maps where they've mapped how signaling goes from your brain to your heart when it comes to electrophysiological signaling, which has implications for how our nervous system functions during disease. This is relevant for all the human genetics data that we study. But our job is to figure out how this is relevant to a broader user base. So our initial focus will be to put the knowledge of all the common fund resources into one place. Basically, one-stop shopping, as I like to refer to our knowledge portal for human genetics. So how are we going to do that? Well, we're going to do that by defining knowledge in a very specific way. Because in addition to being funded as a knowledge center, we're working with an incredibly great group led by Avi Mayan at Mount Sinai, who many of you may know from other settings because he builds really cool data resources. They were funded to be our counterpart in the Data Resource Center. So their job is to aggregate all the raw data from these resources and make that accessible, because there's two layers, right? There's the raw data that all these resources have done, but then there's also the knowledge. So we are really well complemented there to build a single portal ultimately that will share both of these. So if you're a data-driven person, you can access all the data. If you just want the knowledge, you can access all the knowledge. So this will be great. You'll be hearing more about this as we go, but I wanted to just start with how we're thinking of tackling this challenge. So types of knowledge. We've parsed it into 4 different categories. The first being foundational knowledge. What is the hypothesis study? Why are they doing that? Putting that all in one place in a very specific and clear representation that describes what these resources were intended for. This is just an example from GTEx, right? The types of knowledge. primary knowledge. This is basically the basic results that are derived from the study. What did the study overall learn? So for GTEx, here's an example from an abstract. So these are things that would be in the marker papers, basically the flagship papers from all the resources. This is not in one place currently. We think this is incredibly valuable to the resource. If you go to all of them, you can find it quite readily, but bringing it all together so that you even know that it exists is a valuable activity. The second is anything to be drivable from a single common fund data coordinating center, which is what they call these 14 resources, or that's not necessarily published, or pulling together types of knowledge across these data coordinating centers into one visualization. Again, here's an example from GTEx where you'll see all the violin plot results for all the tissues in GTEx. So this are like sort of the derivable results that if you know the resource well, you can probably navigate to. If you don't know the resource well, we want to raise it and make it accessible to you immediately so you can learn the types of knowledge you can derive from these resources. So our initial focus is around all these 4 different prongs: primary, secondary, and foundational. Tertiary is later on in the mix. Our goal with that, the reason tertiary is later, this involves things like I think did I put it in things like knowledge graph results and assertions. This is going to be coming over time, but our initial product will not necessarily focus on those things. To give you an example, very tangible to you as human geneticists, here's the tertiary knowledge. So these are knowledge graphs as well. They're going on within the process. So how are we going to build it? We want to build something that's very simple, very clear right off the bat. And we want to have 3 features. The first will be a landing page. These are some beautiful mock-ups that DK and Alex Sheelan on our team have built to just give you a teaser of what we're thinking. This is really not the focus of today because it may look very different. But the idea is that, there'll be one landing page and it'll be a single search bar, something very simple to what we have done for our other portals, where you can search by entity. I'm going to talk a little bit about what entities mean. Entities are very different in this resource, right? Many of them have information on gene, many of them information on diseases, and that's probably where we're going to start. But then you have things like systems, organs, processes that we believe will be really great entities to allow our user to search by, and then we will string together results for all the knowledge generated on a given CFD resource in one place. But we must do this not just by like puking out the knowledge. We have to figure out what are the scientific questions. And we love to structure scientific questions around the resource we present. So how are we going to do that? Well, again, we think about users. We really do. I mean that because the resource doesn't get used as you're thinking about that. So, for human genetics, we think about the human geneticist or the translational biologist who has a gene they want to study, right? So that's what our portals are built for currently. These resources span the gamut of scientific researchers, right? So some of them are so well focused that we'll never be able to serve them all, of course, but we want to serve the largest scientific community we can about the knowledge within these various resources so that they can, if they choose, dive into one resource they may never known even existed, right? So that's the first one. So how do you do that so it's very clean and clear to someone? We want to present results in the terms of workflows by contexts. So we do workflows now, right? But we want to translate this more into, I am a computational scientist. I would like to have the access to the raw data. So we want to tailor all the experience to that experience. So if you almost filter and customize your view, like you do in many cool websites, like from Amazon or whatever, you customize your view and it will, the page will render like it is thinking like you ideally, right? So here is the access to the APIs. Here you can go download this. This is the information that you would get in this resource from specifically data. These are the data types, the data files, file names, the format, and you can extract those if you want. But many people don't want to do that, right? They don't even know what they're looking for. They don't even know what questions they want to ask. You probably want someone who's, perhaps a medical geneticist or a person working on a target, right? We want to make sure that it's at their hands. And so we want to translate this into, I am a blah, blah, blah person, and allow the resource to present to you information it believes are most useful to you in that context. You still can drill down to these resources, and we still want to make it very clear that if you want to go to Metabolomics Workbench to figure out all that is known about metabolomics in the NIH space, you can go to that resource and find access to the data. So let's talk about how we're going to do that a little bit. So we're hoping that our first product will allow you to search by gene, disease first, and then ultimately system and organ and process as we develop. So give you an example of the type of knowledge we want to represent. Here's a great one that's very useful to you all, right? So oftentimes beyond genetics, you want to understand metabolites, right, for a given disease, trait, or cell type. So Metabolomics Workbench is a CFDE resource. It's been around for over 5 years, I believe. It's incredibly well staffed. It's like, not staffed, resourced. It is the single place that I imagine with most of metabolomics data, like in the world, as we've learned, is deposited. And you, if you want, can go in, search your favorite disease, get all the studies involving, say, diabetes, and you can filter by phenotype, I'm sorry, not phenotype, species, by where it's collected, how many samples, what collection mode, the QC process. The resource has documented all that for you. They are experts in metabolite data, the QC, the processing of it, and the representation of it. They know it better than we do, right? So you as a user, if you know metabolite data, you can select your own data sets and do a meta-analysis and present all of the top metabolites for diabetes curated by yourself with this resource. So that's really great, right? That's incredibly useful. They do this for a host of traits. They have visualizations. It's incredibly useful. But could you imagine a world where you study diabetes and you want to know everything about diabetes from these common fund data resources? Well, the first thing we hope to do is we actually can, there's no way that this is pre-computed right now because that's not what the resource focuses on. What we're going to do is we're going to work with them to have the best practices of what they believe are the canonical analyses for each given trait for metabolite data that they house, they QC, they understand, pre-compute those results. And so you, when you search on the Knowledge Center, and ultimately you will find for your favorite trait, what are the top metabolites associated with that trait. But if you want to go back to Metabolomics Workbench, again, just as we do with our current portals, we make it easy for you to get to that resource, understand the provenance, understand how it's done. So this does two things. It makes it easy for you, even if you're an expert in this resource, but it also elevates the use of this resource ultimately. And also it triggers us to think about other scientific questions you can ask with these data that you may not have even known were possible. So that's what it means for you. What's very cool about this is while we'll also be building a knowledge center that's a standalone resource for all of these common fund resources, all of the results that are relevant to our human genetics portal will now be viewable in that context as well. And that's really exciting for us because it's something that we've heard about for years where people have said, you know, why don't you have kids first data in? That would be really great. They have this amazing wealth of whole genome sequencing data on these rare diseases. Wouldn't it be amazing if you're searching in the portal for your favorite trait or gene for common associations, if you can know what kids first may say about it? We'll now be able to do that firsthand. And this has been an incredibly cool experience because we have gone to each of the common fund resources over the last 6 months and had in-depth conversations with them, just understanding them, their brand, their ethos, their thought process, their consumers, their contributors, and how we would surmise all that and represent it to you. It's been a really fun experience. So a new webinar series is coming in April. I'm going to hand it over to Marie in just a second. And this will be different than our knowledge portal ones for human genetics. We're going to stick to these. We just want to make you aware of it today. But what we're going to be doing with this is all Common Fund data ecosystem members will attend this as well. And they will be giving presentations on a rotation basis about these Common Fund data ecosystem resources. But we will also be representing the unique results and data generated across the Common Fund through our Knowledge Center and the data resource portal that Avi Mayon's team is building. So these will be Fridays at 1 p.m. We picked a time that we thought would be useful because there's not a lot of major webinars during that time, it turns out. So it's really great for our CFDE colleagues. So I will share this with you once it's up and running if you're interested, but I just want to let you know that's going to kick off in April. So with that, I'm going to turn over to Maria, who's going to talk a little bit about these DCCs, just to sort of giving you a teaser of some of the things we're thinking about. And if any time along the way, you think of things like, hey, this would be really valuable to make accessible, feel free to pop in the chat or have a comment.
Okay, let me share my screen. Okay. All right. So I'm just going to take you on a brief tour through all the DCCs, and there are quite a few of them. I'll just say a few words about each one to give you an idea of the breadth of the topics that are included here. So I'll start with 4D Nucleome. This is an organization that is looking at nuclear organization, the organization of nucleic acids in the nucleus in space and time. So it's not only three-dimensional contacts, it adds the dimensions of time and space. And so they have they have many data types, which pretty much boil down to gene expression, chromatin conformation, and nucleic acid interactions. They are also looking at RNA interactions besides DNA interactions. And a couple of their signature tools and features, they have a visualization that some of you may be familiar with called HiGlass that looks at interactions between within a chromosome, between different regions of the chromosome. And try to get my slides advanced. They also have a 4D nucleome browser that lets you browse in a genome browser type view all their interaction data and also gene annotations and other genomic data like that. And also a very cool thing, you can see the 3D conformation of the chromosomes and highlight different parts of them. Okay, so GTEx, which we already mentioned, and I think many of you are familiar with, is another of these data coordinating centers or DCCs. The main part of their data is just bulk expression from 54 different tissue sites in 1,000 donors. They're also starting to look at single nucleus expression data from 8 different tissue sites. And they also call expression and splicing QTLs. So these are variants that are associated with different levels of expression of a gene or different splicing patterns of a gene. They also have some genotypes and genome sequences and some histology images of the tissues that they look at. So they have many beautiful visualizations. Noelle already showed you this one on the left with bulk tissue levels. They also have a really nice one where you can enter a tissue and then get the top 50 expressed genes in that tissue. And not only that, but see the expression levels of those genes in other tissues. So you can see whether they're expressed, they have tissue-specific expression or not. They also have nice visualizations that show RNA levels for a gene in single nucleus experiments. You can also look at expression per exon, not only per gene, and you can enter a set of genes and compare their expression levels. Okay, the next DCC is xRNA. This is something that I wasn't even really aware of before starting this project, but extracellular RNAs are actually emerging to be very important in physiology and disease. And so these are RNAs, either small RNAs or pieces of larger RNAs that are outside the cell, and they're carried in lipid vesicles and bound to proteins sometimes in nanoparticles. So this consortium is just trying to find out how many of these are exist, and what types of RNA, what types of carriers, and which biofluids they are in. So they are kind of starting out here. They don't really have many interfaces. You can, of course, search, filter, and download a lot of data sets from this. And yeah, I should say, just in general, all these DCCs, one of their major jobs is just collect data and data sets and make them available. So all of them have really nice faceted searches for data sets to find a data set with custom criteria. Okay, and yeah, they do have some tools, and this is an example of that data set filtering ability on the left. You can choose multiple criteria to find the data sets that you're looking for, and then you can also visualize lots of different aspects of those data sets with a sophisticated viewer. Okay, HubMap is another one that's been around for a while that you may know. And this has the rather ambitious goal of making an atlas of the whole human body at the cellular level, which of course is critical for understanding disease and health. And so they have a lot of different data from gene expression, chromatin conformation, lots of multi-omics and imaging. And so they're putting it all together to make an atlas. And they have lots of very sophisticated tools. These are just a few of them. There's a human reference atlas where you can look at the three-dimensional structure of organs. There are some different visualizers for single cell data. They have a tool where you can look at histology images and spatial omics in the same context. Okay, so that's HubMap. The next one is IDG, or Illuminating the Druggable Genome. And this one is based on the idea that most successful drug targets are within 3 different protein families, either G-coupled protein receptors, ion channels, or protein kinases. And so they are focusing on those protein families, but they have data from for other proteins as well. So they have this Pharos data portal that really draws, I think it draws from 80 different resources. It's amazing. And they draw all kinds of different data relative to genes and proteins, and present it all in an easy to see display. This is just one part of that. There's this graph showing how much is known in each of the various areas. It's interactive, so if you mouse over it, you can see what these parts of it mean. And the major work that they do at the DCC is to curate the proteins, to curate the information about the proteins, and to classify them in terms of what they call their target development level. So what's the weight of evidence that this protein is a likely drug target? And so their classifications are either, there are some known drug targets, there are some that are suspected because of biology, there are some that are suspected potential targets because of clinical evidence, and then there are some that they call dark, which are just unknown proteins that, because of their membership in protein families, might potentially be good drug candidates. Okay, next one is LINCS, Library of Integrated Network-Based Cellular Signatures. So this one has primarily transcription data, transcriptome data, and what happens to transcription patterns in response to perturbations like drug treatments. And they look at a lot of co-expressed gene sets. So they have a tool where you can find transcriptional signatures, and they've extracted transcription data from not only from their own resource LINCS, but also from GEO and GTEx. And you can search this by a gene or gene set or a bunch of other criteria and come up with transcription patterns that where sets of genes may go up or down in response to this to a treatment. Okay, Glygen is about glycosylation. And this resource just, well, so protein glycosylation is adding a carbohydrate moiety covalently to a protein, and there can be many different kinds of carbohydrates or glycans added to proteins, and this affects their structure and then, of course, their function, and it can have roles in health and disease as well. So it's really important to understand glycosylation, and this DCC has compiled lots of data relevant to glycosylation from many different sources and gathered it all. And they have what they call a super search, which is very versatile, that allows you to make very detailed custom queries. All right, Metabolomics Workbench is all about metabolites. I see we have a question. Maybe I should
I have a question.
take a look at that.
Go ahead, Angie. You have
Yeah.
your hand raised?
I think we have to allow her to speak. Let's see.
Oh, okay, I can do that.
Okay, it should be in the participants list.
You got it. Hold on, Angie, we're coming.
Oh, here, yeah, okay.
Oh, that's okay. Sorry, it was a mistake. Thank you, though.
Oh,
Okay.
okay.
I'm
No
I'm
problem.
appreciating this overview. It's very helpful. Sorry about that.
No
That's
worries.
okay. We've all done it. Okay, so Metabolomics Workbench has just a ton of metabolomics data, and from both targeted and untargeted studies. So targeted studies are where they study specific metabolites that they're looking for. Untargeted means they gather data on everything that's there. And they have some tools where you can, oops, sorry. get back to where I was. They have some tools where you can analyze a data set. For example, with this MetStat tool, you can take a study and generate a report of the metabolites that are in it, and they're color coded by the type of metabolite, and their abundance and so forth is in this table as well. You can also take a study, and they have lots of different tools to analyze it. So you can take the results of one study and look at it in many different types of visualization. They also have a tool where you can take a set of metabolites and see how their structures compare to each other and which are most similar to each other in terms of structure. Okay, MotorPack is another DCC it's really interesting. It's looking at the effects of exercise and doing multi-omics to see how exercise affects all these different processes in the body. This one is kind of just starting out. They are going to look at both rats and humans during or before and after both endurance training and resistance training. And so far, they only have data for rats. They're still collecting data on humans. But so here's just a summary of the data they have for rats. So, of course, with rats, they can get samples of many, many more tissues than they can in humans. So they've looked at all these different tissues on the x-axis, and then they look at many different analytes, as they call them, things that are analyzed, all the way from DNA methylation, open chromatin, transcription levels, and then protein levels and protein post-translational modifications, also metabolites, and it's just really a comprehensive panel of analysis. They have some tools where you can analyze these data. There's one where you can pick different data types. These are just two of them, and look at the data over time by gene for one particular gene. So here's a couple of things for one gene in different tissues. This is expression levels in two different tissues. One really interesting thing from this DCC is that so far what they've noticed is that the patterns in females and males are quite different. And I think that's really interesting. It'll be interesting to see if they see that in humans as well. They also have a visualization, which is tissue level graphical clustering, where you can see how the different analytes change over the time course of the experiment. Looks like we've got
Maria.
yes.
Yeah, I was kind of, it's Ronnie, that is yes, I love your suggestion. In fact, that's something that we've considered. Here's a question. Have you thought about constructing a search feature that takes in user questions, a way to guide the user to the right resource? Wondering if generative AI, yes, brilliant suggestion. We will we need some sort of reward for that. That is something we've been actively thinking about to do. We want to balance that carefully, right? We don't want it to be like chatbot land. But one of the things we've considered is, first of all, actually Alex and DK, who are our UI wizards, have thought about this already. Almost the idea of at the top, you almost build your own story, if you will, based on your context, which is like, you could almost say it's two things. You can imagine a world where you say, I am a blah, 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 and I want blah, 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 right? We give you filters and it guides the user towards that. The other one is type in a question. The questions, I think, require more generative AI, but we thought that we'd start with a few contexts. If you can think of ones that you would like, I'd love to hear them. I know you are actually that type of target user. I could imagine being very useful because you are a human geneticist, but you're also an experimentalist. You think about, these scientific questions related to human disease. So that's a great suggestion, by the way. Any, do you want to comment? Do you want me to unmute you or anything like that? Because I actually love that suggestion. Hold on, let's figure out how to do that. Participants, there's always too many buttons for Zoom. There should be like a chat bot that just tells you. Okay. Ronnie, I unmuted you if there's anything you want to say.
Oops, sorry.
Thanks. Well, I mean, you hit it perfectly. I was also thinking that since you have such dispersed data and organize them might be a challenge. Somebody might just say, hey, I'm interested in metabolomics in women in these maybe contexts of disease. And the search can help people get to the right data that they might want to find quickly, because it sounds like there's gonna be a lot of information that's gonna be, instead of like searching through a bunch of different clicks and options, this could be a starting point.
Yeah, it's a great suggestion. It's actually something I think that Avi's team is thinking about. They are thinking particularly because their orientation is really about, I want to get at the data and actually like extract it, or actually just know what the data says, not necessarily like the distilled knowledge, which we're hoping to get at on top of that. But he is thinking about very cool, he calls them his playbooks, which allow you to stitch together exactly what you said, metabolites, women, diabetes. And there's a set of cards that use generative AI to pull together what DCCs, data coordinating centers, or the resources have information on the things you're asking. And that allows you to at least just see that at a glance. But I know over the next 5 years, he's thinking of going deeper, which allows you to actually like extract the data. So this concept is going to work both for the Knowledge Center and for the DRC. And I love that suggestion. I see other things in the chat. Sorry, I'm going fast. Questions. Let me take a look at these and I will, why don't you keep going, Maria? I don't want to stop you.
Okay. Yeah, so I hope everyone's getting an idea of how diverse these resources are. It's really amazing. Another one is called Kids First, and this looks at children who have either childhood cancer or structural birth defects, and I guess there's an idea that they might have some shared genetic architecture there. And so primarily what they have is sequence data. They're doing whole genome sequence data on these kids and their parents. And they also have, they're doing RNA-seq and exome sequencing in tumor samples. And they're also importantly taking all the phenotypic data about the patients and harmonizing them so these studies can be compared. And so they don't really have any user interfaces at the moment, but they do have all the data, and authorized researchers can get access to these sequence data and work with them. Okay, Spark or stimulating peripheral activity to relieve conditions, is about bioelectronic therapies, which I also wasn't really aware of. I guess that using electronic therapies for neurological conditions is a really promising area of research, but we don't know enough about the autonomic nervous system to really to do that. So this resource is aimed at making an atlas of the nervous system. And so they have electrophysiology data, single cell transcriptomics and imaging. And they have some platforms where you can look at computational models and simulations, or integrate graph-based data. And then Noelle showed a picture like this before. This is a different one. It's just they make these wiring diagrams of the nervous system and how the signals go between different types of cells and how they end up at organs. So this is really complex and interesting. All right. A2CPS is another one that's just getting going, so they don't have a lot of data available yet, or user interfaces. But this is based on the idea that when people have acute pain, like knee pain, and they have surgery for it, some of them recover and are fine, but others transition to having chronic pain in that site. And it's not clear why, and how to predict who that would happen to. So they are looking at patients with either knee surgery or thoracic surgery, and measuring their outcomes and doing lots of other measurements on them, like multiomics. They're also testing their pain sensitivity and things like that, looking at their biomarkers and doing neuroimaging of their brains. So this should give a lot of data along with the patient outcomes of whether they did transition to chronic pain, and that should be very illuminating. Okay, we're almost at the end here. SenNet is about cellular senescence or aging, and they want to identify the types and locations of senescent cells in the body in different states of health, and basically make atlases of them, as very similar to what HubMap is trying to do, but focusing on senescent cells. And so yeah, this one is also kind of a relatively new one, and I don't think they have much data available at the moment. And finally, one that is really just starting, Bridge to AI. So this is they're trying to generate data sets that will be AI ready and figure out what it takes to make a data set AI ready. And so they have 4 data generation projects which are starting up that will generate this AI ready data. And that's about all. It's really just starting at the moment. So that is the whirlwind tour of the DCCs. And I don't know if we have more questions. Oh, no, else typing an answer. That was all I had for today.
That was just a very quick tour, fire hydrant like of it was actually, it is a lot for us to take on. So our MVP, as we like to call it, is going to be very simple. It's really about, in fact, as we've talked to the DCCs themselves, they're a consortium of consortia, where they have meetings, annually or semi-annually, where they get together and they talk about their own work. And even amongst this consortia, they aren't aware of all the things that are going on. And that's part of the reason why the NIH funded the data ecosystem first in its first pilot, just to get the data types. So there's a really cool effort. They have this model already within them called the C2M2 model, where it's a metadata model. It's how they share metadata and data across the ecosystem already. So there's already really nice guts and innards, if you will, for connecting data across these resources. There's also elegant APIs in many cases. So it's really ripe for integration, which is really, very exciting. This is something that we haven't encountered before. And as we've learned, these resources, they're magical in some of the things they do in terms of visualizations. And, as folks I mentioned earlier, building portals, this makes it really exciting for us to learn from them all and then help elevate them. So, that's kind of where we're headed. So as our user base here, we're hoping to hear from you. Ronnie, that was a great suggestion. Eva, that was a really great suggestion, thinking about diabetes and thinking about MotorPack. Again, MotorPack is one exactly, studying metabolism. MotorPack will be very valuable to our common metabolic disease user base because they have genomics, they have omics data, they have it well processed, they have it in one place. And we can find ways to represent that in a resource that summarizes what they're learning about genes affected by exercise and different conditions over time. This is really great for interventions and therapies, and useful for all of human disease context. So that's one that we're excited, specifically with our background in common metabolic diseases. In other places that's really been excited, you find that different disease communities have relationships. So HubMap has a strong relationship with some of the folks working in the pancreas sector, just because they have commonalities in some of the visualizations for Pancreas Atlas. So this is also relevant to us as a resource. So we're very excited about this. It's a big challenge, and we're stepping out of our comfort zone. So we really want to just make you aware of it. Next month, I think we're going back to our usuals. So features and focus groups on different things we're working on. Something coming on the horizon from our internal common metabolic disease knowledge portal is that we are debuting a tissue-based view of the resource in March. And we will also have a lot of development around registering your own data as we develop our, bring your own research results feature that we've built over the years. We want to enable anyone to bring more genetic and genomic data to both the Common Metabolic Disease Knowledge Portal and to the CMDGA. So we want to have an interface that makes it super easy for you to upload your results, configure pages so that you can view the results yourself, and then contribute them to the knowledge portal. So that's something you can hear about. We heard about CoLocus a couple weeks ago. CoLocus will be something that we'll be featuring in the spring as well, integrated fully on the portal. It's there as a labs page now. What else is coming in March, Maria, that we want to mention to the group in terms of the knowledge portal things?
We'll have new data sets, as always. But I think you covered all the features.
Okay, very good. All right, well, with that, any other questions? Is anything else in the chat that's new that we didn't cover?
Yeah, these were just observations from Eva. Thank you, Eva.
Comments about when Elon Musk makes it into your webinars, that's when the man has penetrated every sector of the world. What will this guy do next? My. All right. Well, it was a pleasure. Thank you for listening. I know this is kind of a new thing and maybe not as directly relevant to your research, but you know, it gave us a chance to speak about it. It's the first time we've spoken about this externally. So for me, it was actually fun to kind of think about what are the most salient things to share with your user base. So again, feel free to give us feedback for these things and any ideas for webinars as you go forward. And we will share the public webinar for the CFDE. It's going to kick off in April on these channels and through our user base from the knowledge portals for common disease. These will feature, the first one's going to feature, I believe, which DCC is presenting. Mackenzie would know that, but she's on vacation. But anyways, it's going to be a DCC is going to speak about what's most important to them. And then the DRC, Avi's team, is going to speak about the products they're building. And then we will talk a little bit about the Knowledge Center. And these will be monthly and feature all these DCCs. So if you're interested in any of them, you can join. And again, same type of structure as these are. So with that, I'll give you back your 9 minutes. And thank you for your time and attention. Thanks, Maria.
Thank you. Thanks, everyone. Bye-bye.